Protocols and tips about Western blot by cyan. Firstly, I'd like to reiterate the biological hazard about the reagents used in Western blot. We use PMSF as protease inhibitor when preparing protein samples. We use the lysis buffer Triton-X, and we use SDS, DTT to maintain the reduction state of the proteins. Arquebus, AP, and timid are used in preparing the gel. And sodium azide, we use it to inhibit bacteria, or we use it to prepare the stripping buffer. They're all toxic, so be careful and protect yourselves. This page showed a short history about the electrophoresis used by the scientists to detect different hybridizations about the DNA, RNA, and proteins. Several kinds of controls are used in Western blot, such as the internal reference control, like the actin tubulin and gap DH and etc., the positive control, and the negative control, and the blank control. We use different internal reference controls according to the location of the proteins we'd like to detect. Don't choose an internal reference control similar to the molecular weight of your target protein. The reference protein is often an important enzyme or cytoskeleton. For example, GAPDH is a key enzyme for glycolysis, so it cannot be used during diabetes or hypoxia. I use the samples on the right to illustrate how to interpret the experimental results. Both lanes used liver cancer cell samples. After MG132 was used to inhibit ubiquitinase degradation, the redox factor NRF2 was not degraded, so it was detected. The gap DH at the bottom indicates that the sample amount is the same. There are several steps in the process of Western blot: protein extraction and quantification, electrophoresis, membrane transfer blocking, primary antibody and secondary antibody incubation, ECL development, and membrane regeneration. These are the principles of the protein extraction. Use the lysis buffer according to the protein distribution. Remain the dissolved state of the protein. Use the protease inhibitor and phosphatase inhibitors without inducing chemical modification. Sometimes you can remove high abundance protein and nucleic acid interference when necessary. Remember, do not freeze and thaw repeatedly. This page shows that we can use different lysis buffer plus differential centrifugation to obtain proteins in different subcellular localization. Here is the principle for protein extraction in different subcellular location. We use different lysis buffers and centrifuge at different speed and different temperatures to get their supernatant or their precipitation. Important to mention, the uniformity of the sample ensures the repeatability of the subsequent experiments. Here are some protein quantification methods. In our lab, we often use the BCA method. We make a standard curve with the serially diluted standards and calculate the concentrations of the sample according to the curve. If the sample concentration is out of the curve range, the sample can be diluted appropriately. For sample preparation, add sample buffer to the protein extraction, boil it at 99 centigrade Celsius for 10 minutes, cool it immediately after the high temperature denaturation. For gel preparation, different concentrations for SDS page have corresponding optimal separation ranges. Use fresh, AP, and protected from light. As to the electrophoresis, use 20 to 50 microgram total protein per well, and the volume and the concentration of each well are the same. The empty lanes on the both sides are loaded with loading buffer. If your protein samples have very small molecular weight, as the autophage marker LC3 or the nucleus internal reference histone S3, you can only electrophoresis to half the sheet of the gel to prevent horizontal spread of your samples. On the contrary, if your protein sample has the molecular weight similar to mTOR, you can let the bromophenol blue run out a little of the gel. There are two common ways for the membrane transfer, and the first is semi-dry and the other is the wet transfer. After transfer the membrane, we can confirm it through pre-stained protein marker or other stains. For macromolecular proteins, 0.1% SDS can be added to the transfer fluid to help transfer. Decrease methanol to 10%. Consider wet transfer overnight at 4 centigrade Celsius. For small molecular proteins, SDS prevents the protein from binding to the membrane, so SDS can be removed. For blocking, we can use 5% skimmed milk powder or 3 to 5% BSA. Keep it in room temperature for 1 to 2 hours or 4 degrees Celsius overnight. There are monoclonal and polyclonal antibodies for us to choose from. If our sample species is A and the primary antibody species is B, and we should use the secondary antibody species to anti the primary antibody. The advantages and disadvantages of the chemiluminescence method and chemical coloration method of the development are listed as above. If your protein sample are very precious, you can consider use the membrane regeneration. Just shake it in straight buffer at the room temperature for 10 minutes to an hour and reblocking, and then followed by the next steps. It is worth mentioning that if your membrane generation goes not thoroughly, the final result of the development may seem very similar to your last result. On the contrary, if the membrane regeneration goes too thoroughly, the development of the protein amount may be below your actual protein amount. Due to the limitation of the video length, I put the supplementary reading materials to this page. There is a brief review about the choice of protease and phosphatase inhibitors, the function of different components of lysis buffer, and comparison of applications of different lysis buffer. Thanks for watching. If you enjoyed this video, just replay it. If you have any questions or suggestions, feel free to contact me through email. If there is the next episode, see you then!